Really, the bottom line for this is that you don't know what you have until you test for it. And that just by checking a box on the submission form doesn't mean you're using the best test for your situation or for the organism's biology. So for years, we had been testing for Pneumocystis murina and never found it, but that didn't exactly mean it wasn't there. And it's possible that low prevalence organisms, which don't transfer easily to our soil bedding sentinels, like Pneumocystis murina, are just lurking in our colonies.
Hello and welcome to The Last Word, the official AALAS podcast dedicated to advancing laboratory animal science. I'm Jennifer Smith, Chief Scientific Officer at AALAS, and also your host. Together, we'll discuss current research, emerging trends, and the innovative work that supports scientific progress with animal welfare. We are so glad you're here. Let's discover something new together. Welcome back. In this episode, I am joined by a fellow colleague, Dr. Lauren M. Habenicht. And Lauren is a senior clinical veterinarian and associate professor of pathology at the University of Colorado Anschutz Medical Campus. She received her DVM from Colorado State University and completed her lab animal residency at the University of Washington. And she is also a diplomat of AACLAM. Lauren is joining us today to discuss her recent JLAS paper titled Detection and Remediation of Pneumocystis murina Infection in Environmental Health Monitoring. Lauren, thank you so much for being here. I'm so excited to hear more about you and the paper. So go ahead, tell us a little bit about yourself.
Well, thank you so much for having me on. This is exciting. Well, I'm one of those veterinarians who wanted to be a doggy doctor since before I could say the word veterinarian. I also around the same time wanted to be a ballerina and an astronaut, and preferably all three at the same time. I took a detour through classical music, really focused on that in middle and high school. I went to an arts magnet school. I played and still play the flute quite a bit. But I became interested in biomedical research as a teenager after my dad developed kind of a strange Parkinson's disease-like syndrome. And it progressed really quickly while he was still pretty young. And after he died, I realized just how much we had yet to learn about medicine and especially about the brain. And I found that I couldn't relate as well to my colleagues at the art school who really had a singular focus on improving their art form and perfecting their craft. And I found my mind wandering to science and medicine a lot more often. And I started college thinking that I'd probably do an MD, PhD, become a physician scientist and hopefully a neurologist. But by third year of college, I'd kind of come back around to veterinary medicine. I was a little disillusioned by the pre-med culture and had some great experiences in research labs working with non-human primates and got some pre-veterinary mentoring from some of the titans in our field. I didn't realize who they were at the time and what a big deal that was. Having Dr. Jim Fox around to talk about veterinary school and the veterinary career was pretty incredible. Still, by the time I applied to veterinary school, I was still thinking I'd go into small animal general practice, maybe be involved in some clinical trials or something, still contribute to science that way. I was fortunate enough to get accepted off the wait list for vet school in my home state of Colorado. Go Rams. And partway through my first semester, I was already tired of memorizing the textbook. I know vet school's improved a lot since then, which is great. But I sought out some more experience and joined a lab in vet school, working with the postdocs there, as well as the veterinarians that helped take care of the mice and the cats that we worked with. I realized that lab animal really encompassed all of my favorite parts of veterinary medicine. I loved thinking through the complex physiology of internal medicine cases. I loved thinking about the population health and keeping large groups of animals healthy in shelter medicine. And I was increasingly drawn to exotic species as well. So being able to contribute to biomedical research while still serving as an advocate for all the animals that make those discoveries possible. Really, the career is like a win-win-win, wins all the way around. And so I charged through residency in comparative medicine right after vet school, and I haven't looked back.
Wow. I loved hearing that, Lauren. Thank you so much for sharing all of that. I really appreciate it. And I know that must be tough. And I want to also say I'm really sorry to hear about your dad. That must have been a really difficult time And I'm just so thankful that you were able to kind of use that experience and help sort of shape you. And wow, we are really thankful that you're in this industry with us and partnering and doing such amazing things. I love it. And I love how you describe just kind of how you got here, because I think we all have a really unique story, and I really appreciate you sharing yours.
Thanks. I'm so glad I found the field because it's been a fantastic experience so far.
Oh, that's awesome. And I loved hearing about the flute. I think that is amazing. I think we could have a talent show at some time.
Ooh, yeah, let's do it.
I would love
AALAS
it.
national meeting. We can strike up a band.
Yeah, we could be like the APV band. But I appreciate that. And I think you just really embody lab animal medicine. And I just love hearing everything about your story. Okay, well, let's jump into the paper, since that's why we're here. We always ask our authors to talk about the team, because we know it takes a team. And this was a really unique paper and case series, really, I guess is what we should call it. And so I'd love to hear more about the team and kind of who did what and all of that.
Sure. So our author list is comprised of a bunch of veterinarians. I have some excellent colleagues at CU Anschutz. There's a team of 5 of us vets. My fellow senior vet, Dr. Mike Fink, helps run our import-export program. So as someone who runs the rodent health monitoring program, I work really closely with him to figure out exactly what we should be testing for, how we report that, so that we can help researchers share mouse models across the country and across the world. Dr. Derek Fong is the Associate Director of Veterinary Services for the Office of Laboratory Animal Resources here, and of course oversees all the veterinary care. So when we're dealing with sick mice, he's an important member of the team. Dr. Chris Manuel is the Senior Associate Director of our Office of Laboratory Animal Resources, and he's really the person who oversees biosecurity in our animal facilities, and he served as a mentor to me throughout this process. And of course, our director and attending veterinarian, Dr. Jory Lazinski, was a huge support throughout this, helped make sure we had all the money to do all the testing that we had to do as we chased down these 3 cases. We also worked really closely with our colleagues at IDEXX Bioanalytics, Dr. Sherry Hamilton and Dr. Marcy Hart. They shared their expertise in the tests and our sampling strategy, as well as spent a lot of time reading and interpreting histology slides as we were evaluating our response to Pneumocystis murina on campus. So they were a huge help. Everyone helped write the paper and or revise the manuscript of that group that I listed. But I also want to give a shout out to our fantastic tech team, both the vet techs and the animal care techs, who made sure that we could actually get all of this done.
Oh, thank you so much for sharing all of that. And I love that you were so detailed, because I think sometimes folks that want to do research and especially write up these manuscripts and get them accepted are, you never really know, like, who do I need to have and how many people and all those things. So thanks for giving that such a detailed description there. We love it. And of course, you have a great team there. So that's exciting.
Yeah.
All right. So tell us, how did you get interested in this project? Why this project? Kind of what spurred you guys to think, let's talk about getting this written up?
Yeah, well, it definitely fits into my interest in population health that I mentioned before. So I help run both our rodent and our zebrafish health monitoring programs on campus. And in early 2022, we switched to 100% environmental health monitoring. We had been using for many years, had worked with a soil bedding sentinel system. And so we relied on serology testing, looking for antibodies to indicate that there were any rodent pathogens in our rodent colonies. So now, since 2022, we are doing PCR testing. So we're looking directly for the nucleic acids that come from those pathogens. And I think probably the biggest fear making that switch is that you might find something that you thought you've been excluding and maybe thought you've been excluding for many years because you've had lots of negative tests. So being able to share our experience with this, what happened when we did find something unexpected, how we dealt with it, and how it worked out for us, I think has value for others as they're thinking about making the switch away from soil bedding sentinels, and help provide a little guidance for someone who might have gotten a surprise positive, especially for a respiratory pathogen like pneumocystis.
Oh, here. I thank you for sharing that too and really teeing up what we're going to talk about. And I think this is such a timely paper, which is why we picked it too.
Yeah.
I always want to learn as well. And it's such a timely topic. And it also is one where you're kind of on the other side of it, right? Your institution has made the commitment to not have soil bedding sentinels. And so it's so great to hear kind of this case series of what happened afterwards. So we appreciate that. All right, so let's just jump into the introduction. You do a great job of describing our new little friend,
Yeah.
our new little agent, or I guess I shouldn't say new, right? But it would be great to just have you unpack some of that introduction because it's quite good.
Sure. So pneumocystis is a fungus, and it's an opportunistic respiratory pathogen. It's species specific, meaning that though humans, mice, rabbits, rats can all develop pneumocystis infections, they're all caused by different species of the fungus. And so rats can't spread it to mice can't spread it to people, etc. I described it as an opportunistic pathogen, meaning that healthy animals, or people, say, generally don't get sick if they're exposed to pneumocystis, and it takes either genetic or medically caused immunosuppression for the infection to really take hold. Affected mice may initially present with weight loss, hunched posture, ruffled fur, and that can progress to respiratory signs like labored breathing or even death. Although we didn't necessarily see a lot of these signs, so it wasn't really high on our radar. We weren't that suspicious that it was happening in our colonies before we got our positive tests. Because of the number and the variety of immunocompromised animals that we have on our campus that are involved in cancer research and a lot of other translational projects, it's really important for us to protect animals from opportunistic infections like this. And we understand that mice with normal immune systems can clear pneumocystis murina after they're exposed and not get a chronic infection. But there's a lot less known about mice that have modified but not necessarily deficient immune systems. In the paper, we call these immune vague. So they may have changes to a specific type of white blood cell, but they still have most or all components of a complete immune system. And we have lots of these immune vague mice on campus, many different types with different small alterations to how their immune systems work. These models can help scientists better understand how the individual elements of the immune system either help or hinder the body's response to disease. And it also means that these strains of mice can have different immune responses to mouse pathogens that we may learn about in school. We know that many different cell types and immune signaling molecules called cytokines play a role in the body's response to pneumocystis, which can make it difficult for us to predict if any particular strain will or will not get infected. And because pneumocystis can make some mice sick and it can affect research outcomes, it's excluded from most research colonies, or so we think. As a respiratory pathogen, it's not very efficiently detected by soil bedding sentinels, which, as I mentioned, has been kind of the mainstay of many rodent health monitoring programs for decades. So I think it's possible that there's a lot more of this out there than we might think. And so that's why we wanted to share our experience with the lab animal community.
Oh, that's great. Thank you for summarizing that so well. And yeah, it was a good reminder for me in reading through that just about, how far we've come with environmental or the duct sampling as you guys talked about it. I really love the immune vague term. I think it's funny, but it's a perfect term. I love it. And I think that is such a good way to describe so many of the models that we work with now. They are immune vague. And so you don't really know what bucket to put them in. And it sounds like your program is not unique in that you have so many, and being charged with protecting them from these things pathogen wise is really important. All right. Well, I guess this is such a great story on these cases. So I guess just dive in. Tell us kind of how one case happened and then kind of how it springboarded into the others. We'd love to hear more.
So I'd like to start by telling you all a little bit about the structure of our rodent health monitoring program and some of the equipment that makes it possible. So we test for a group of viruses, bacteria, and parasites quarterly. And for efficiency, we test for a smaller group of pathogens 3 quarters out of the year. And then once a year, we test for a much longer list that includes some less likely but still important pathogens, intending, right, to confirm that none of these are present in the colony at detectable levels. And I said it that way because unless we're using a perfectly sensitive test and directly testing 100% of the animals in the colony, we can't guarantee per se that an organism doesn't exist in the colony, but we can make a very good educated guess based on the design of how the test works and what our testing strategy is, so that we can really accurately represent the health of the entire colony based on our sampling. We have over 22,000 mouse cages across several facilities on campus. And of these, 99%, maybe a little more, are individually vented cages with filtration at the rack level. So that means that air from all cages on the rack carry particles from everything that lives in those cages, from the mice all the way down to their microbiome. And that air goes into the rack's exhaust system, where those particles can either stick to a little piece of exhaust dust media that's positioned in the exhaust, and some of them can fall out of the air and kind of stay in the plenum or the air tubes in the rack. For the rodent health monitoring program, I take advantage of this engineering to get a pretty complete picture of what organisms are or are not present in the up to 70 or 84 cages that are on that rack. If a rack tests positive for, say, pneumocystis, we can triangulate which rows have affected cages by testing those plenums or the air pipes in each rack that test positive, and which cages on any of those rows that are positive might have mice that are exhaling pneumocystis organisms, with additional environmental swabs and PCR testing still. So all of these positive cases that I'm going to talk about came up during the first round of our annual panel. So for many years, we had not seen any serologically positive sentinels for Pneumocystis murina. And so we figured that was pretty unlikely to be in our colonies. And so we only tested for it once a year. I'll say that changed after we started finding it once we had our environmental health monitoring system in place.
Yes, I'm glad you added that little footnote.
So when we made the switch, we kind of went cold turkey. We thought the evidence was there that environmental health monitoring can be very sensitive, probably more sensitive than our soil bedding sentinels had been. And so we were ready to just pull off the Band-Aid and make the switch. That said, we started with our shorter quarterly panels for the first 3 quarters. And luckily, there were no surprises during those first 3 quarters. So I was very pleased about that.
Is that the time when you're just waiting for that email and you're...
Yep, exactly. When you see the email come through.
Yeah.
And
white knuckle on your mouse, waiting for the email, right? Yeah.
And finally, it was time to do that full annual panel. So yeah, same white knuckle on the mouse, opening that email.
It's time for a quick break to hear about the AALAS Learning Library. Hey, are you ready to elevate your team's knowledge and skills in laboratory animal science? Well, the AALAS Learning Library is your one-stop solution for comprehensive training. With courses designed for everyone, you can train your entire team in one place, earn valuable AALAS CEUs and RACE CE credits, and take advantage of group discounts to maximize that investment. Getting started is easy too. Sign up for free to access select courses instantly to explore a vast library of resources. Empower your team, stay ahead of the field, and grow with the AALAS Learning Library, your trusted resource for laboratory animal science professionals.
In that first round, we've found in one of the facilities, case number 1 in the paper, we had 3 racks out of a larger room with about 14 racks total. 3 racks in that room came back positive. So before freaking out, the first step was to confirm that those were actually true positives. So get some more exhaust dust and send those in. We got another month's worth of dust on those racks and then resubmitted and found just 2 of those 3 were true positives. Both of those racks housed mice from the same research lab, so that seemed to make some sense. And so from there, we narrowed down which rows on those 2 racks and then which cages were positive, working with the lab to understand if those cages were related, if they had anything in common. And we found that only mice from a single strain tested positive by PCR, serology. We even did histology on a few. They were basically 100% of the cages of that strain that the lab had. These cages were not clustered together on the rack. They were kind of all over the place. The lab had used every space available to them on their racks, and so they filled them in wherever it worked. But we were happy to see that none of the adjacent cages tested positive if they were a different strain. So from that, it seemed like the nose-to-nose spread within the cage was probably what was happening. Could have initially come from a chronically infected breeder pair. They had a single founder that they started this colony from that they'd imported years before. And we were happy to see that it seemed like the normal cage and animal handling practices we had on campus and thorough use of disinfectant seemed to be enough to contain the spread of Pneumocystis murina. So overall, that was pretty good news. If you have to have this bad news, this is pretty good news.
Right.
Yeah. This strain, also good news. This strain had, since the lab imported them through our import-export program, the strain had since become available commercially. So the solution in case one for this group was basically to wrap up studies with the mice they had of this strain and then order new mice from the vendor that the vendor had commercially re-derived. So we didn't have to worry about pneumocystis from those guys. Of interest to me at the time was that this was not an immunocompromised strain. So when I think of Pneumocystis murina, as I talked about in the intro, it really affects immunocompromised mice. So if these were NSGs or nude mice, then I'd be more concerned. And so I wouldn't have necessarily suspected that these immune vague mice would have gotten it. They did have a limited T cell repertoire. So, they definitely had some changes to their immune system, but it was not a strain that I would have thought would develop this chronic infection and be able to pass it on for generations and generations, because it had been years since the strain originally was imported into our program.
That is interesting.
So the second case presented a little more of a mystery. We used the same testing and confirmation strategy, found a single rack in a room in a different facility that was positive. There were a couple of different labs mice housed on that rack, but we used our plenum row swabs and then our cage swabs to identify just a single positive cage on the entire rack. Those mice had been bred in-house and the breeders were still around and the breeder cage wasn't positive. So we weren't exactly sure where that came from. And unlike case one, where the mice were immune vague, this was a strain with just a GFP reporter, so it should have had a normal immune system. As this labs project was wrapping up anyway, it's possible that there could have been some other positive cages previously, and that might have contributed to some of the DNA we found in the rack, but it was kind of exciting to see that our testing method was sensitive enough to be able to identify just a single positive cage. Remediation in this case was that the project was ending. So the lab collected the rest of those mice and it was all taken care of and done. So fortunately, it's not always that simple, right? So the first two cases, I was like, oh, that's not too bad.
Very true. Right. Very true.
So the third case we had was a little more of a challenge. In one of our smaller housing rooms in one of the facilities that has 7 single-sided ventilated caging racks, all 7 of those racks tested positive the first time we tested for Pneumocystis murina. Some of the racks had 1,000 or more copies of DNA, and in the other ones we were looking at maybe up to 100 or so. So this was red hot positive. Some of the racks in the room, however, had less than 10 copy numbers. So again, a little more equivocal. We saw immediately that this was a Pneumocystis murina infection on a much larger scale than the other 2 cases. Based on the copy numbers and which researchers had mice on which racks, we suspected that the positive racks with low copy numbers, like in the tens and teens, could be false positives. And that due to the design of the racks and the amount of pneumocystis present in the room with those racks that had 1,000 copies or more, that DNA could just be kind of floating around in the air and getting into the rack and onto the media, even though we didn't think that any of the mice on those racks could be or were likely infected. So as we worked to identify which cages and strains were true positives in the red hot racks, we also moved the suspected false positive cages to their own room with clean cages and clean racks, new media that hadn't been exposed to any of the air in the original housing room. In that separate space, we did 3 rounds of monthly follow-up testing in the media on those new racks. And we confirmed that those were false positives. So we never got any positive tests once we moved those racks and those mice out of the room with lots of pneumocystis in it. Meanwhile, we found that there were multiple immune vague strains in the high copy number racks that were positive. So interesting here, we found that some cages could test positive once and then negative a little bit later, maybe sometimes come up positive again in the future, or vice versa. We also saw that offspring of a Pneumocystis murina positive breeding pair would usually test negative around the time of weaning and for the next month or two, but often around month 3, we would see that those guys would turn positive. So their cages would have enough of the Pneumocystis DNA for us to be able to detect it with our PCR swabs. It's kind of just an observation at this point. We didn't really look at the kinetics of infection in these guys, but just something for others to keep in mind before using this type of cage level test to rule in or rule out pneumocystis from a colony. So you might want to wait until they're a little bit older and have had time to develop the infection and shed. So in case 3, this third case, we did 2 rounds of cage level testing before deciding whether we could consider one of these strains actually pneumocystis negative. So if all cages from that strain were negative both times, and we swabbed the cages and cage components right before cage change, so they had the highest chance of containing pneumocystis DNA, if they were always negative, then we felt confident that those strains were truly negative. And so once we identified those, and it was just 3 in this case, to reduce their risk of exposure, those strains were moved into another room where, of course, we continued to watch them really closely and perform rack level surveillance. But luckily, those never tested positive. And then we were left with 12 strains from this lab that tested positive at some point during the confirmatory tests. All cages of these strains were considered positive. So even if we just had 1 or 2 cages of mice from one of those strains, we just considered that all of them likely were carrying or were really susceptible to it and could become positive at any time. So we kind of kept those all together. As I mentioned earlier, we didn't see a lot of clinical evidence of Pneumocystis murina in these strains. The lab had some challenges breeding. They had a couple of these strains had slightly shorter lifespans that seemed to kind of decline earlier than other strains. But again, like no smoking gun to say, oh, this is likely Pneumocystis murina. We didn't see respiratory signs. So, but because they had some issues breeding and we knew that there was a lot of pneumocystis, we decided to start treating these mice with a uniprim diet, which contains therapeutic levels of trimethoprim sulfadiazine. And that's known to minimize disease symptoms, even if it hasn't been proven to clear a pneumocystis infection, but could help support the mice and potentially just reduce the amount of pneumocystis in the room. So basically our plan, and it worked out pretty well, I'll say, it was just that we were going to treat the mice until the study ended, which there was an end date in sight for this group as well. And then the colony shut down while the mice were still being treated. But they were 8+ months on the uniprim diet.
Wow, that is such a great description of those 3 cases. Thank you. I have so many questions for you, which I know you addressed in your discussion. So we're going to kind of morph to that part. But thank you again for kind of describing all 3 of those scenarios are so different and unique, yet this kind of underlying agent. And I really appreciate, and I know our listeners will too, of just sort of the thought process from your team of what do we do? And then I also really appreciate that you're including just sometimes we do have to pivot when the, investigator is done with the study or doing something else. They pivot, we pivot. Mm-hmm. That's what makes lab animal medicine so fun, right? And makes us good at what we do is just that ability to pivot. So, and I love how you included that because it did alter maybe ultimately what you did in some of those cases. And so I think that's really important. So I guess let's just jump into the discussion. However you want to do it. You want to case by case or just generally? You have so much knowledge here. We'd just love to hear more.
Sure. Well, I can kind of give a general wrap up and
Sure.
then we can, go question and answer from there if you want.
Sure. Sounds good.
So, I mean, really the bottom line for this is that you don't know what you have until you test for it. And that just by checking a box on the submission form doesn't mean you're using the best test for your situation or for the organism's biology. So for years, we had been testing for Pneumocystis murina and never found it, but that didn't exactly mean it wasn't there. And it's possible that low prevalence organisms, which don't transfer easily to our soil bedding sentinels, like Pneumocystis murina, are just lurking in our colonies. So this is yet another good reason to switch to environmental health monitoring. I know we've been talking about that a lot on the podcast with Dr. Wei Hansen as well.
Yes.
And even if you don't make the full switch, just adding it to the program for specific pathogens, because it can be more sensitive for some, might be a good idea. So I talked a little bit about PCR copy numbers, using those to interpret our test results in case 3, but they were an important consideration in all 3 cases. I wish I could say that below a certain copy number on your test results, you don't need to worry about it. It's probably a false positive. Unfortunately, it's not that simple. If there were just a few affected cages, like the one that we found in case 2, the PCR results could look similar to the false positives that we saw in case 3. So the only way to know really is to conduct follow-up testing.
I really appreciate, I was just going to interrupt you there, but I really appreciate you saying that, because I think if you're new to environmental health monitoring, testing this way, you might think that there is some sort of quantitative way to say, above copy number X, you got a problem. Below copy number X, don't sweat it. Of course, I'm joking, but I really appreciate that you said that, because I think, at least for this agent, it doesn't sound like that's the case. And I think that's important information to know.
Just give your finance team a heads up that you're submitting more samples.
Another sage piece of advice. Yes.
Right. I've had people ask about our cage swabbing strategy. So why not just test the mice directly? And there's actually a publication about the success of using oropharyngeal swabs of rats to test for their species of Pneumocystis. However, when we were chatting with our colleagues at IDEX, Dr. Hart had looked into this previously and found that, at least in their lab's hands, for mice, testing the cage environment was more sensitive than doing the oropharyngeal swabs, possibly because of intermittent shedding. I mean, that's what I think we were seeing in case 3 when we'd have cages that were positive and negative and positive again. Swabbing the cage is also a lot less disruptive to the mice. We're opening their cage to swab the lid and swab the walls versus a brief restraint for the animals and putting a swab in the back of their throat. I think we can all relate to that not being particularly pleasant after COVID.
Yes, very good points there. Yes.
So with our caging type and the PCR tests we were using, at least environmental swabbing seemed to work really well for us, but we didn't directly compare it to oropharyngeal swabs. So that still could be a good way to do it, depending on the circumstances.
Yeah, I appreciate you saying that too, and especially in light of when they're shedding and there's no overt way to pick up a mouse and say, "Are you shedding?"
Right.
So
cough for me.
Yeah, right. So I appreciate that. These subtleties are really helpful. So yeah, keep going. What else?
Well, one thing that I'd love to know that we weren't able to test in this process was if treating the Pneumocystis murina infections with the trimethoprim sulfadiazine diet could be effective at actually clearing the infection in these immune vague mice. We saw that after several months of treatment and a rack change, our exhaust dust media was consistently testing negative for case 3. But we never challenged the mice by, say, ending treatment and then just waiting to see if the pneumocystis would come back. But that's what people had seen in immunocompromised mice in the past. So that's why we don't think that it cures pneumocystis infections. But I wonder, with a slightly more functional immune system, if mice can actually clear it with a little support. So that would be good to know, especially for trying to manage this in the future. One big challenge with that, though, is that all of these immunovague strains are a little bit different. They're all unique. So even if we had found that it worked in one or a handful of our strains, that wouldn't really guarantee that treatment would cure all immunovague mice. I did, at the end of the project for case 3, I did perform necropsies on a dozen or so of the mice that we'd been treating for months. And We looked for pneumocystis by PCR of the lungs and also in histology and didn't find any in those animals. But again, that's not strong enough evidence to claim that it was actually eliminated from the colony with the treatment and wouldn't have come back.
Yeah, that's interesting. All right. Well, and unfortunately, we are getting to the end, and I really have enjoyed this so much, and I know our listeners have too, because you've just shed a lot of light on an interesting topic, which is, once you make that commitment to environmental sentinel sampling and then you get a positive, what do you do, so to speak? So I just love this case series because of that. And I love that their cases were also different. All right. So if you've been listening to our podcast, you know that we ask one final question of our guests, not very original. But the question today for you, Lauren, is what advice would you give somebody who wants to improve their detection or remediation of P. murina in their colonies? Like, what would you say to them?
Well, I don't think any of us really want to detect a pathogen like this in our colony.
Good point. Good point.
Especially something we think we're excluding. So in an ideal world, right, we do lots and lots of intelligent, smart testing, and all of those tests come back negative. That said, I kind of want to give a quick shout out to one of my residency mentors, Dr. Susan Dowling, who was a very early adopter of environmental health monitoring over a decade ago. As a resident, I remember I would see her light up and be excited when a test came back positive. And at the time, I thought she was nuts. I was like, nope, that will never be me. Positive tests are bad news. It's super scary. But I have to admit, I've kind of come around. Now, of course, I'm very happy with negative test results in our health monitoring program, but I also do get a little bit excited when there's an unexpected positive that needs to be investigated. So thanks, Sue, for showing me that that's possible.
Yeah, that's great. I love that. That's great.
But I guess my advice is for everyone to really work to optimize your health monitoring program to maximize the chances that you'll be able to detect the presence of pathogens that could have a significant impact either on your research program or on the health and welfare of the animals. And as you chatted about with Dr. Wei Hansen, that could look different for different institutions. So we're an NCI designated cancer center with lots of immunocompromised mouse models. And we also have a really large immunology department with many immune vague mice. So by improving our ability to test for opportunists like pneumocystis, murina, that really feels like a must for our program. So I'm glad that we made the switch, even though it ended up with a lot of extra work for me. But I think the key word is optimize. So you don't need to test for more agents or test more often necessarily. You just have to figure out what works for your institution. And for me, probably one of the scariest parts of making the switch to environmental health monitoring was the possibility that we'd find an organism we thought we didn't have, which is basically exactly what happened, right? But by sharing a few different real life scenarios of how we handled it and lived to tell the tale, hopefully we can empower others to make the switch with a little less apprehension. Now there's a little bit of a framework, something you can base your response on. And so you can also effectively manage the unexpected positive pneumocystis murina test or anything else without big depopulations of animals or major research disruptions.
Oh, what a great summary. And I just really love how you summarized that from fear of getting a positive to turning it into kind of like, okay, this is a project. And I really do appreciate exactly what you said, that your team has really provided for us a framework of, hey, this is how we handled it. And it doesn't have to be complete depopulation necessarily. And I really appreciate that. Oh, well, this has been so great, Lauren. I really appreciate you coming on here and the work that your team has done. And I hope that we hear more from you, although I hope that it's not a lot more positives in your future.
I hope not too, but you never know. That's why we test.
Thanks for joining us on The Last Word, the official AALAS podcast dedicated to advancing laboratory animal science. We really hope you enjoyed this episode. To find out more about this guest or the conversation, download our show notes wherever you access this podcast. And please join us for our next episode. Until then, stay curious and stay connected.